Welcome to audio paper series with Yasir from Quadram Institute, Norwich, UK. This week, I'm talking to Dr. Emma Holden from Quadram Institute about her recent publication of Massively Parallel Transposon Mutagenesis Identifies Temporally Essential Genes for Biofilm Formation in Escherichia coli. It's published in Microbial Genomics in 2021. Emma, would you like to introduce us to the project, please?
Of course. So this project looks at what genes are involved in biofilm formation in E. coli over time. So when we study bacteria in the lab, we normally study them in planktonic culture swimming around. And that's not how bacteria normally exist. They exist as part of these biofilms, these structured aggregated communities of bacteria. And biofilms go through this life cycle where planktonic cells attach to a surface, they grow and mature, and they produce this extracellular matrix to form the mature biofilm. And a lot of work has been done on characterizing the mature biofilm, but my work looks at what genes are involved throughout the life cycle, what's involved in adhesion, the process of maturation, as well as the mature biofilm. And we've investigated it using TRADIS Express, which is this massive high-throughput transposon mutagenesis technique, which identifies the genes involved in survival under stress.
Brilliant. So what were your observations when you were studying biofilm formation?
So we found that there were 48 genes involved in biofilm formation in E. coli throughout the biofilm life cycle. So there were some genes that were involved at each stage that we looked at. We looked at after 12 hours, 24 hours, and 48 hours. There were some genes that were involved at all three stages that we looked at. They were involved in fimbriae regulation, such as FIM-E and FIM-B, and genes involved in motility, such as LRHA and TOM-B. We also found that as the biofilm matures, it has different requirements. For example, in the early biofilm, genes involved in adhesion and motility were important. And over time, genes involved in matrix production and purine biosynthesis were involved as the biofilm matured. One very interesting thing that we found is that there are some genes, such as DSBA and DKSA, where their effect on biofilm fitness changes over time. So for DSBA, it's very important in the early biofilm for adhesion, but in the late biofilm, its expression is detrimental for biofilm matrix production. So we have this temporal essentiality for these genes. And the last thing we found is that we found 5 genes that had not previously been implicated in biofilm formation. And these were ZAP-E and TRU-A, involved in cell division, MAO-P, involved in chromosomal organization, and YIG-Z and YKG-J, the functions of which we don't know yet.
Interesting. You mean different sets of genes are required at different stages in life cycle of biofilm?
Exactly. Not just one size fits all.
That's a very, very good, very nice observation. So what can we do with this observation? What's the application of this observation?
So these genes could be used as targets for anti-biofilm drugs or targets for pro-biofilm drugs. Biofilms are detrimental for hospital infections, but they're also beneficial for creating drugs like insulin. So we can target these genes to either benefit or destroy the biofilm. And we can also use them as biomarkers. If you pick up expression of these genes, if you pick up production of these gene products in the blood or the sputum or any sort of diagnostic material, we may be able to identify biofilm infections from these biomarkers.
Lovely. Thank you very much.